Good evening, students. A warm welcome. And today we're going to start a new series, which is Rapid Revision Program, where we have taken high weightage chapters, and we're going to do a one-shot videos based on that, really taking the important topics of that particular chapter. As you can see, today we're going to start off with molecular basis of inheritance. So let's dive into the class then. So as you know, molecular basis of inheritance has got a connection with our first year chapter, that is biomolecules. So one should have a thorough knowledge of biomolecules before you guys start off your molecular basis of inheritance. Apart from that, our cell cycle and cell division do also have link with this particular chapter. And in terms of weightage, okay, 6 to 8 questions is what one can expect from this particular chapter. Clear? So it's a very high weightage chapters. So what are we going to see today? We're going to obviously talk about replication. We're going to talk about the transcription. We're going to talk about the post transcriptional changes. We're going to talk about the genetic code, and we're going to talk about translation, and we're going to talk about gene expression, which includes our lac operon. So these are the topics that we're going to cover today. Let's see what we have in the store. Let me ask a question before I say anything. What is DNA? What is DNA? And this is how usually I start my class whenever I start either biomolecules or molecular basis of inheritance. And most of the students say, sir, DNA is nothing but deoxyribonucleic acid. But this explanation, even 6-year-old, 7-year-old, nowadays can also say what is DNA. But DNA is not just deoxyribonucleic acid. So what am I expecting from you? It is the genetic material. Then my immediate question for you guys will be, what is genetic material? My immediate question for you guys will be, what is genetic material? So genetic material is a substance which stores, carries, and transfers the information from one generation to the other. So this is how you can define genetic material. So what is this substance? This substance is nothing but DNA. This substance is nothing but DNA. But what do you mean by information? Where does the information lies? The information lies the information lies in the sequence of the nucleotides. That is what the information is all about. So what do you mean by that? Can you elaborate? Can you elaborate the this particular sentence? For example, AUG carries a certain information, GUG carries a certain information. For example, AUG codes for an amino acid called as methionine. GUG codes for an amino acid called as valine. For example, AAA, can anybody say for which amino acid does it codes for? AA codes for an amino acid called as lysine. AGA, anybody? AGA codes for an amino acid arginine. So as we change the sequence of nucleotides, the information changes. So that is what we mean by the information, right? So DNA is a substance, and rather the genetic material is a substance, and that substance is nothing but DNA, which stores the information and carries throughout the lifetime and transfers it to the next generation. Clear? And the information lies in the sequence of the nucleotides. The information lies in the sequence of the nucleotides. So let's now go back and see. So deoxyribonucleic acid and ribonucleic acid are the two types of nucleic acids found in living systems, and obviously predominantly DNA is the genetic material in most of the organisms. RNA though act as a genetic material in some viruses, for example TMV, for example QB bacteriophage, but mostly the RNA functions as messenger. For example, mRNA functions as a messenger. Adapter, your tRNA molecule acts as a adapter. Okay, structural and catalytic. Structural, obviously rRNA, which forms our ribosomes, is a structural RNA. Catalytic RNA, rRNA again. So which particular rRNA? For example, 23S rRNA acts as an enzyme, which we call it as ribozyme. What is the function of this 23S rRNA? We'll see that in a bit. Okay, so let's move on. So DNA is a long polymer of deoxyribonucleotides and is negatively charged, and this negative charge is imparted to the DNA by the moiety phosphoric acid. So phosphoric acid that forms, or that is the important moiety of the DNA, imparts negative charge onto the DNA molecule. The length of the DNA usually defined as the number of nucleotides, and which is a characteristic feature of a particular organism. See, you can see over a tabular column over here, which really explains about 4 different examples, and each of them have been asked in the previous exams. And one more thing that you need to make sure that you concentrate, that you can see it is the haploid content of human DNA in the question. So if you ignore whether he is asking haploid or diploid, then you'll choose the wrong answer. So just by reading n number of times NCERT, we always recollect 3.3 into 10 to the power of 9 base pairs. But sometimes, okay, question can be asked even the diploid human DNA, which is 6 into 6 into 10 to the power of 9 base pairs as well. One has to remember these data. Very, very important. Let's talk about something about DNA packaging, where questions have been asked in the previous years. So all of us know that there is, okay, for example, imagine this is our cell cycle. This is your M phase, this is your G1, this is your S, and this is your G2. So over here, you know the genetic material in the G1 is present in the form of chromatin. Over here also it is present in the form of chromatin. Over here also it is present in the form of chromatin. But over here in the M phase, you will see a highly condensed form, that is your typical chromosome. So this is your highly condensed form, that is your typical chromosome. So even though it looks like a, like a mesh or something like who has dropped like that, but there is an order even this structure called as chromatin. So even within chromatin as well, there is a sense of packaging involved. There is a sense of packaging involved even within the chromatin as well. But if I ask a question, is packaging involved in prokaryotes? What is your answer? Yes or no? Is there any packaging involved in prokaryotic DNA as well? Yes, there is a packaging, but it is not as elaborative as packaging in the eukaryotic cells. So there is a packaging involved, and yes, there are proteins involved as well. So there are proteins involved as well in the prokaryotic DNA packaging. It is not scattered. It is not scattered. It is not scattered in case of the prokaryotes. Okay, everybody thinks, sir, can you see like this? So usually you will see, okay, the DNA of a prokaryotic cell like this. So they think it is scattered. No, it is not scattered. It is, okay, properly packaged. Yes, compared to that of eukaryotic cell, you don't see such high level of packaging. It looks more or less like this. There will be a center protein scaffold. There will be a center protein scaffold to which the DNA is arranged in the form of the loops. The DNA is arranged in the form of the loops like this. So this is what, okay, we see in terms of prokaryotic DNA packaging. What are these? These are the loops. What loops? DNA loops, which are attached to the protein scaffold. Okay, so there are proteins involved, but these are not histones. These are not histones. But let's concentrate on our eukaryotic DNA packaging. So DNA packaging in eukaryotes is of high level. In eukaryotes, it is much more complex. That's what I've been saying. It is carried out with the help of basic amino acids or basic protein rich, okay, one called as a histone. So it is histones or positively charged proteins which are rich in amino acids like lysine, arginine. So genetic code, we have 61 codons which codes for amino acids. So it is difficult to remember the entire table as such. But whenever possible, if you can learn few codes, for example, everybody knows the start and stop codons. But apart from that, if you can remember, for example, lysine is coded by a codon, one of the codon, lysine is coded by AAA, arginine is coded by AGA. So if you remember these sort of stuff. So it's much easier for us to remember the genetic code. So histones are positively charged proteins which are rich in basic amino acids like lysine and arginine. Okay, guys, are you following me? Next. So these histones, okay, on their own, okay, you don't see them. You will see the histones forming a structure called as a nucleosome to which the DNA is wound. To which the DNA is wound. For example, okay, in a typical nucleosome, you see 8, which is nothing but octamer of histone proteins. Octamer or histone proteins. So what are they? They are H2A, H2B, H3 and H4. So you said octamer, but there are only 4. So H2A1, A2, H2B1, B2, H31, H32, H41, H42. So there are 1, 2, 3, 4, 5, 6, 7, 8, to which the DNA is wound around. To which the DNA is wound around like this. And these ends are packed or closed together by a clipper, which is your H1 histone. H1 histone, which acts as a clipper. Okay, it ties the both ends of the DNA. And what is the length of the DNA that is wound around a nucleosome? Okay, roughly about 200 base pairs of the DNA is wound around the length of to one particular nucleosome. The length of the DNA that is wound around the nucleosome is of 200 base pairs, and H1 acts as a linker or as a clipper, and which is one of the largest histone out of all these histone proteins. Okay, so this is a typical eukaryotic chromosome organization. So all of you know what is this. This is your typical metaphase chromosome structure. And if you stretch it further, you get a structure called as chromatid. And if you stretch it further, okay, you'll get a structure called as a supersolenoid. If you stretch it further, you'll get a structure called as solenoid, which is of 30 nanometer in diameter. And then if you stretch further, you'll get the, what do you say, this is your typical nucleosomes. But if you stretch it further, that's when you'll get a typical double helical structure. But if you stretch further, nothing will be there. Okay, you can't stretch it further, the structure will collapse. So this is your high end, highly condensed form of the chromosome, and this is the least condensed form of the genetic material. So if you stretch it one by one, this is where we end up. Once the cell division is completed, what will happen? This DNA wounds around the nucleosome, forms a chromatin and forms the solenoid, further condenses, forms supersolenoid, forms the chromatids, forms the chromosome. So either you go this way or this way, you can better understand the eukaryotic chromosome structure. Clear? So you'll not get any questions from this particular point. I just picked up for you guys to understand how actually eukaryotic chromosome structure organization is done. Okay, and when the cell is not dividing, you see the genetic material, you see the genetic material, you see the genetic material in the form of chromatin. When the cell is not dividing, for us if you see the nucleus, it looks as if, okay, someone has dumped the DNA, but believe me, there is a organization even in so called messy mesh like structure over here. So imagine if any foreigner comes over to India and if they see our traffic, they go insane. They say there is no order, but we Indians know how to maneuver around. So we know there is a order in the disorderliness as well. So as superficial look, it appears as if it is a mess. There is no orderliness over here. Believe me, there is an order even this superior messy like structure, and this chromatin is divided into two regions, that is euchromatin and heterochromatin. Euchromatin is lightly stained, loosely packed, and transcriptionally active. Transcriptionally active. Whereas you see heterochromatin, this is darkly stained, this is densely packed, and this is transcriptionally inactive. So, can we identify by looking at mere diagram of a chromatin which one is euchromatin and heterochromatin? Yes, it all depends. Okay, while you are drawing, you make sure you draw like this. So, this highly entangled region over here is usually our heterochromatin. Over here, this is our euchromatin. Okay, shall we move on? So let's talk about replication. Let's talk about replication. So I'm not going to simply read out what it is there on the slide, which you guys can do. Let me pick up something which is there, okay, in the form of explanation for you or in the form of questions. So as all of you know that DNA replicates semi-conservatively. Semi-conservatively. Yes, everybody knows. But let me ask a question. Who proposed? Who proposed the semi-conservative mode of replication? Who proposed? Sir, it's a no-brainer. The answer is Matthew Messelson and Franklin Stahl. Okay, if this is your answer, you're absolutely wrong. They didn't propose, okay, the semi-conservative model. It is the Watson and Crick who proposed. Who proposed is your Watson and Crick. but it has been experimentally proved by these two gentlemen. Hello, is it clear? So, sir, how can you say that? But if you see under the DNA replication, there is a particular statement that they themselves says that we are aware. In principle, we are aware how DNA replicates. How DNA replicates in principle, they do have an idea which is clearly mentioned in their own research article. In their own research article, they mentioned that we're aware, మాకు తెలుసు, okay, ఎలా జరుగుతుంది, how it happens, we do know how it happens అనేసి చెప్పడం జరిగింది. So be careful what is being asked, who proposed or who proved. Okay, that should be your answer accordingly. So it has been proved by Matthew Meselson and Franklin Stahl in an experiment, okay, with the help of E. coli, and this has been proved by Taylor and et al. Taylor et al. Et al అంటే ఇంకొక పేరు కాదు, టేలర్ మరియు ఇతరులు, Taylor and others. So Taylor and et al. Has proved that DNA replicates in a semi-conservative mode even in plants, and the plant is Vicia faba. So in this plants, they have proved that DNA replicates in a semi-conservative mode. DNA replicates in a semi-conservative mode. So everybody knows this experiment. So let me, okay, tell you the important thing. Here small numerical kind of questions will be asked. What is it? Obviously, they'll ask after so many generations, if I've grown in a heavy medium and then in the light medium, what will be the number of hybrid DNA molecules? What will be the number of the light DNA molecules? So let me give one thing over here. Easy asking, number of hybrid DNA molecules, or is he asking percentage? So these two things you need to concentrate. For example, let's see over here. Okay, when I grow the E. coli cells, both the strands are of heavy. When I grow these cells in a light medium, I'll get both of them as hybrid. After next generation, I'll grow them in the light medium again, so I'll get both light and hybrid. See over here, what is the percentage of heavy? 100%. What is the percentage of heavy here? There is no heavy anymore, you have only hybrid, so you got 100% hybrid. We got 100% heavy over here, we got 100% hybrid over here, we got 50% light, and we got 50% hybrid. So what will happen after 20 more minutes? What will happen? The percentage of light DNA increases to 75 and the hybrid DNA will decrease, okay, by 25%. Next, again 50%. Again it decreases. So it keeps on decreasing, but the number of hybrid DNA molecules will remain 2 even after many generations. See here, if percentage is being asked, it decreases. Here it is 50-50. Next generation, the light DNA increases because we are going in the light nitrogen medium. So what decreases? The hybrid decreases. What increases? The light DNA molecules increases in terms of percentage, but the number of hybrid DNA molecules, even after 20 minutes, I mean 60 more minutes, I mean 20 this 20 40, another 20, that is 60. Even after that, you'll get only two hybrid DNA molecules. It doesn't change. Please do pay attention over here. The number remains same, whereas the number of hybrid DNA remains same, whereas the percentage changes. So let's go through quickly what is replication is all about. Okay, so let's see what is replication. Duplication of the DNA. Duplication of the DNA is called as replication. So let me start off replication by asking a question: in which phase of Streptococcus pneumoniae DNA replicates? In which phase, okay, it occurs? First, G1 phase, second, S phase, third, G2 phase, before fission. So please answer this question. Meantime, I'll grab some water. Okay, so let's see how many of you answered right. Let's see how many of you answered right. So there are many people who has answered, okay, S phase, S phase, which is absolutely wrong. The right answer is before fission. The right answer is before fission, because Streptococcus pneumonia is a bacterium, and you know bacteria are prokaryotes. In case of prokaryotes, no cell cycle. No cell cycle in case of prokaryotes, and it happens just before the fission. So just before a bacterium divides into two, okay, the DNA will be duplicated and it will be distributed equally to the daughter cells. Okay, and it was a previous year question. Okay, instead of Streptococcus pneumonia, I believe he asked E. coli somewhere around 2018-19. So this was a question. So be careful. Just don't jump to a conclusion what they are asking for. Clear? And there is some naughty students who are there online chatting. Okay, they are wantedly saying, okay, A as the answer. I know you are making fun of it. Okay, but it is not the answer. Okay. Let's see how exactly DNA replication occurs. Let me see the enzymes that are involved. The first and foremost enzyme is DNA polymerase. The first and foremost enzyme is DNA polymerase. So DNA polymerase is also called as DNA dependent DNA polymerase. DNA dependent DNA polymerase. DNA dependent DNA polymerase. So imagine this is our DNA. Okay, and this is our DNA. Okay, so which runs anti-parallel to each other and which are complementary. Over here, so for example, imagine, let me grab a book. Imagine this is a book. So imagine this is a book and I duct tape it all the four corners and give it to you to make a photocopy out of it. Obviously, one has to open it in order to do the photocopy, isn't it? So, in order for the DNA polymerase, imagine this is our DNA polymerase, to carry out the replication or the duplication process, the DNA has to open up. The DNA has to open up like this. The DNA has to open up like this. But seldom it happens like that. DNA can't be open in its entirety. DNA can't be opened in its entirety. DNA can't be opened in its entirety. DNA will be opened. So it can't be opened like this. Rather, DNA is opened in a localized manner. DNA is opened in a localized manner like this. So it is opened in a localized manner. And there is a direct statement in the NCRT why it can't be opened up in its entirety like that. So the answer for this particular question is, okay, to open the DNA in its entirety like this, we need high amount of energy. High amount of energy, okay, which the cell is not ready to spend on. So this is one of the important reason. And the second reason, which is not mentioned, okay, it loses the fidelity. Okay, imagine if DNA is open in its entirety, what would happen? What would happen? Nucleases will be eagerly waiting to chop off the DNA. So if you chop the DNA, what will happen? The information is lost. Imagine you open up the door and sleep. What will happen? You are inviting thieves into your house. కదా తెరిచిపెట్టుకొని పడుకుంటే పొద్దున్న లేచి చూసేసరికి మొత్తం వెళ్ళిపోయి ఉంటాయి. So you invited them. So you can't open the DNA. The first reason is, which is written within the NCRT, that high amount of energy is required, okay, which the cell is not ready to spend, and it loses the fidelity, nijayati, okay, information కోల్పోతుంది. ఎందుకని? Okay, it is prone for nuclease digestion. సో ఈ కారణాల చేత ఇది ఓకే పూర్తిగా ఓపెన్ చేయలేదన్నమాట. am I clear guys? Hello? And next, that means somebody else has to come and open their DNA, because DNA polymerase can't open. So somebody else will come, and who comes up and opens the DNA? There is an enzyme called as DNA helicase. There is an enzyme, okay, which opens the DNA, called as DNA helicase. So it unwinds the DNA. Okay, so this is opened up by an enzyme called as helicase. Okay, helicase will open the DNA. Still, who is waiting over here? DNA polymerase is waiting over here in the wings to start the replication process, but it is not ready yet. So what is the problem? Has helicase opened up, okay, there is so much of attraction between these two strands, they rewinds once again, they join once again. Imagine, okay, ఇద్దరు స్టూడెంట్స్ ఉన్నారు అనుకోండి, ఫ్రెండ్స్ మాట్లాడుతూనే ఉన్నారు కంటిన్యూస్గా. సో ఏం చేస్తాము? అరే భయ్ నువ్వు లేచి ఆ చివరి కూర్చో, నువ్వు ఈ చివరి కూర్చో అంటాం. ఓకే, టీచర్ ఉన్నంతసేపు వాళ్ళు కూర్చొని ఉంటారు. అలా జస్ట్ as the teacher leaves, they'll come and sit together and they continues to talk together. So this is what happens between these two strands, that they rewinds together. So in order to prevent the rejoining, we take the help of proteins called as SSB proteins. Called as SSB proteins. What are these? SSB. So SSB. What are these? Single stranded binding proteins. What is the function of them? Okay, they prevent the recoiling of the DNA. They prevent the recoiling of the DNA. Okay, now we'll say why don't the DNA polymerase opens up and carries the function of the replication, but DNA polymerase have two problems over here. DNA polymerase has two problems over here. What are those? Those problems are DNA polymerase have got two problems or two shortcomings. రెండు కష్టాలు ఉన్నాయి, రెండు ప్రాబ్లమ్స్ ఉన్నాయి, దో దిక్కతే హై ఇస్కో. क्या भाई? First one is it can't initiate the process of replication. తనంతకు తానుగా మొదలు పెట్టలేదు. Okay, on its own it can't start the process of the replication. Just like us, imagine you are stuck somewhere, okay, while solving a problem. Okay, you go to the help of your teacher. Then teacher just gives a small hint and they say, sir, please inkekku cheppakandi, I know how to do it. So that's how DNA polymerase behaves. It can't initiate the process of replication, but if somebody comes and shows how to do it, it will do the process of replication. What is the second problem? It can add nucleotides. It can add nucleotides only in 5' to 3' direction. Only in 5' to 3' direction. So as you know, we have DNA strands, okay, in two polarities. Okay, so this is your 5' to 3'. This is 3' to 5'. So it can't initiate the replication. Then what is the solution? Sir, yes, you gave us, okay, that it has problems. There are two problems, but what is the solution? The solution for this particular first problem is in the form of primase, which adds primers. Clear? And what is the solution for this one? That it can add nucleotides only in 5' to 3' direction is, let's see how it overcomes that problem of adding nucleotides only in 5' to 3' direction. So primase is the enzyme which adds primers. So now tell me, in which of these two strands DNA polymerase will not have any problem, okay, in terms of this thing? So 5' to 3' direction. So which one? In which strand DNA polymerase will not have much of a problem? This or this? Okay, this. So if this is 3 prime to 5 prime, it starts off with 5 prime. So there will be a primer that is added because polymerase don't know how to start the process of replication. So it adds primer. Who? Primase adds primer. And then comes our superhero who is waiting in the wings for a very, very long time. Who is it? DNA polymerase. It sees how it is being added over here and now extends the strand like this. Is it clear? Hello? So this is the solution for this problem. But here 5 prime to 3 prime, then it becomes 3 prime, but DNA polymerase says no, I can't go in this direction. I always go in 5 prime to 3 prime direction. So the solution for this one is that we add, okay, n number of primers by creating small false loops so that the DNA can be replicated. Over here there is a primer, and then DNA polymerase comes and extends the DNA like this. So now it becomes your 5 prime, this is your 3 prime. Next comes one more primer over here, one more primer over here, and then comes your DNA polymerase and extends the DNA in this particular direction. Okay, guys, are you following me what I'm trying to say? Clear? So these are the problems that we can overcome. But do remember that we have opened the DNA in a localized manner. So I have opened the DNA in a localized manner. But if I open the DNA in a localized manner, it creates negative coiling. Negative coiling, and there is a chance that the structure might collapse. So you need to release the stress. You need to release the stress. Relieve or decrease the stress created due to localized unwinding. Okay. For example, imagine, ఓకే, మార్క్స్ తక్కువ రాలేదనో మీ ఏదో అల్లరి చేశారనో మీ మదర్ పూర్తిగా లెఫ్ట్ అండ్ రైట్ ఇచ్చారు. బట్ ఆఫ్టర్ 1 or 2 hours she'll come back, beta ऐसे नहीं, okay, she'll pacify, isn't it? So that you don't feel way too bad by giving some, okay, 100 or 200 rupees, chalo, okay, kuch mangalo khalo, but you know it's for your good sake. So అలా ఎప్పుడైతే హెలికేస్ ఇలా లోకలైజ్డ్ మేనర్లో ఓపెన్ చేస్తుందో స్ట్రెస్ అనేది క్రియేట్ అవుతుంది. దట్ స్ట్రెస్ ఈస్ కాల్డ్ as topological stress, which is released or relieved by an enzyme called as gyrase. So who decreases the stress? An enzyme called as gyrase. Gyrase, which is a kind of topoisomerase. This releases your topological stress that is created due to localized unwinding. Guys, am I clear what I'm trying to say? Hello? So let's go back and see over here. So the machinery and the enzymes you need to follow. So helicase, okay, opens the DNA. SSB, okay, single stranded binding protein, prevents the recoiling. Next, topoisomerase. Topoisomerase releases the topological stress, or you can say it decreases negative coiling. Negative coiling. Okay. Next, what is RNA primer? Everybody should remember RNA primer is a ribonucleotides. Ribonucleotides. So, as far as we remember, we are talking about DNA replication. How come you add RNA into the DNA? అరే భయ్ ఒక చిన్న సామెత చెప్తాను ఇక్కడ, గుడ్డి కన్ను కన్నా మెల్ల కన్ను మేలు అంటారు. ఇన్స్టెడ్ ఆఫ్ గోయింగ్ బ్లైండ్, అట్లీస్ట్ విత్ ఎ స్ప్లింటెడ్ ఐ యూ కెన్ సీ, రైట్? So DNA polymerase can't initiate the process of replication, so somebody is coming for the help. So let's take the help, continue the process, and later on we remove them. So primers will be removed before the termination of the replication. Clear? So let's see what is leading and lagging strand. So since DNA polymerase can add nucleotides only in 5 prime to 3 prime direction, so the other strand will be left over, for which see here we are making DNA in shorter fragments. So this is your leading strand, this is your lagging strand. Many of the students think this is the leading and this one is the lagging. No, both are templates, so you can't say leading lagging there. So the one which is continuously being made is called as your leading strand, and the strand where it is made in smaller fragments is called as lagging strand. And this lagging strand fragments are also called as Okazaki fragments, are also called as Okazaki fragments, because Reggie Okazaki is a scientist who has identified these fragments for the first time. So who is it? Reggie Okazaki. And there are many kinds of DNA polymerases like DNA Pol 1, Pol 2, and Pol 3. Pol 3 is called as the major polymerizing enzyme. Major polymerizing enzyme. Apart from polymerization, polymerases do carry out proofreading or editing function. Proofreading or editing function. అంటే యాడ్ చేసిన తర్వాత కరెక్ట్గా యాడ్ చేశానో లేదో తిరిగి చూసుకుంటుంది అనమాట. జస్ట్ లైక్ ఇమాజిన్ యూ కంప్లీటెడ్ యువర్ ఎగ్జామ్, బట్ యువర్ టీచర్ సేస్ స్టిల్ దెర్ ఇస్ సో మచ్ ఆఫ్ టైమ్, వై డోంట్ యూ రీచెక్ వన్స్ అగైన్ whether you have properly written or not. So that function carries out by the DNA polymerase as well. So apart from adding nucleotides, it also adds the other functions, that is proofreading or editing function. Roughly about 2000 base pairs are added per second. So that's how fast it does the work. So over here, imagine this is a strand running from 3 prime to 5 prime, and this is our 5 prime to 3 prime newly strand that is being made. This is your primer, and imagine the nucleotide sequence over here is A T T T G G C C A, like that. So now DNA polymerase extends this primer by adding, if A is there, complementarity T, A, A, A. Okay, obviously C like that. Let me ask a question. So, is this C alone added? Is cytosine alone added? No. What is being added is the DNTPs, deoxynucleotide, okay, or I'm sorry, nucleoside triphosphate, deoxynucleoside, not nucleotide. If I say nucleotide, phosphates are already included, so I can't say nucleotide. So deoxynucleoside triphosphates. So what are those, sir? DATP, DGTP, DCTP, and DTTP. So now let me ask a question. If G is there, if G is there, okay, obviously C will come. That means DCTP will go and bind there. Is it true or false? Is it true or false? Do you see, okay, the entire DCTP being added like this over here? No. No, it won't. Okay, in the textbook, Baba says in the textbook that these, okay, substrate, these are called as our substrates. It has got dual function. Who? This DNTPs has got a dual function. That means they go and add as a substrate, but okay, but they do provide energy. They do provide energy. So what do you mean by that, sir? What do you mean by that? For example, let's see. This is your sugar and this is your base. What is this? Okay, imagine this is cytosine and these are your phosphates. Yes, while being made, it is being made as a nucleoside triphosphate, but when it is supposed to go and add, okay, as a complementary base to the guanine, these terminal 2 phosphates are broken down. These terminal 2 phosphates are broken down. With the energy, it forms the phosphodiester bond over here and attaches to this with the help of 3 hydrogen bonds with the opposite strand. Does it make sense to you guys? Clear? So these DNTPs has got dual function. Very, very important NCRT lines. Okay, what is that? It acts as a substrate and they do provide energy. They do provide energy for the formation of the bond. Okay, am I clear? So the entire process of DNA process can be studied under 3 different headings called initiation, elongation, and termination. So obviously DNA polymerase can't initiate. Who comes to the rescue? Who comes to the rescue? Primase in the form of primer. Okay, it is the DNA polymerase that carries the elongation, and termination is carried out by terse sequences, so which is not there in our NCRT. I'm not going to discuss, but let me end the lecture with a question. Okay, which of the which of the strands complete their replication first? Replication first. So that is your, is it leading strand or is it going to be lagging strand? Is it going to be a leading strand or a lagging strand? Hello? So we thought of taking it as an entire molecular basis in one lecture, but as you know, okay, we are way past to the time, so I don't want to hold up my team. I will continue the next molecular basis, the remaining topics, as a molecular basis lecture 2 part. Okay, so please, okay, please, okay, maintain consistency in terms of your preparation. Okay, so many students are coming and seeing me, sir, we are working so hard but marks are not coming out. So what is happening? Okay, what should we do? I say, be honest, be honest in terms of your preparation. Are you giving, are you giving your 100%? Are you giving your 100%? Are you doing all the four subjects every day with a plan? If you don't have a plan, as I repeatedly says, okay, failing to plan is planning to fail. So have a plan. Even now you can, okay, work it out. If you're consistent in terms of preparation, honest enough, am I being honest in preparation, am I giving what it takes, then everything falls in line for you over the week, or rather over the days. I can't even say now over the weeks now, so it's very close by. So over the days, okay, all the things will fall into the place. Okay, so next class I'll definitely go beyond 1 hour. I'm sure it will take 2 hours. So within the next class, the 2 hours, I'm going to complete the entire molecular basis of inheritance. So please do write your full length test. Many students, I'm talking about my students whom I'm teaching offline, please come back and write the exams. Okay, writing online is not going to help you. Sitting, okay, on the bench every afternoon from 2 o'clock to 5 o'clock is very crucial. Doing the entire four subjects in a stretch is what helps you out. If you are writing exam online or at home, you'll do biology, take 10 or 15 minutes break and come back for physics, and take another 10 minutes break and do chemistry. It's not going to work out. Please, okay, come to the campuses or wherever you are going, sit, okay, or from a stipulated period of time, 2 to 5, my friends, okay, in one single stretch, give the exams. Okay, so I'm losing out marks, I'm scared. Are bhai, if you don't give exams, how do you estimate where you guys are playing? Okay, so it's very important. If our Indian, I know, cricketers thinks that we'll only play, okay, in the nationals, we don't do the net practice, what will happen? What will happen? So imagine, okay, these part tests and these grant tests are very important for you to assess your performance. Okay, thank you very much for joining, and I'll get back to you with the other lecture by the completion of the molecular basis of inheritance. Thank you.